8 mutations and 10 deletions within the gene describe greater than 80% of the Rett girls. And so there are four common types. There's a missense mutation, it's a typo. There's a nonsense mutation that stops the transcription prematurely. And then there are these deletions and sometimes duplications in the gene. Each one of them lends itself to different approaches. So for example, the world is on fire right now. The Nobel Prize is out there for this new CRISPR technology, which is a bacterial antiviral technique that has evolved in bacteria that recognizes viral DNA and destroys it. And people have now adopted it into the human genome, where you can find a single typo, missense mutation, and use technology to go in and splice out the missense and put in the right sense. And so this is one technology that's being applied to certain genetic diseases. The nonsense mutations, where you have this premature stop in the middle of the paragraph, which changes the meaning of the story, it changes the meaning of the protein. There are drugs which can be given which cause the transcription machinery that's reading the gene to skip over that X or that period and transcribe the whole protein. Those who have this X at the end of their mutation, they make varying degrees of the protein depending on where the mutation is. Some farther toward the end, almost complete MECP2 is made and they're much less severely affected. Those at the very beginning, of course, don't make any protein and they're much more severely affected. Again, the deletions and duplications are a third technology that can splice out pieces of DNA, not just a single thing, but pieces of DNA. So we're going to see different strategies evolve